Okay, so we just talked about epigenetic markers and direct differentiation, and now we're going to talk about something called predictive reprogramming. So as we mentioned, as we were talking about direct reprogramming, it's difficult to predict what exact kind of transcription factors are going to cause the differentiation you want. In order to predict this, we need to get the genome, the transcriptome, of the gene of the cell that we want. Let's do that. Let's draw that. Right, so we want cell we have. Right. And you get the transcriptome, right? So all of the messenger RNA. And we get that information and we determine the epigenetics of the cell we have. mRNA. And the mRNA, this one. Right, mRNA, those two. And we use something called predictive reprogramming. And so what that is we use computer analysis to predict what transcription factors would allow us to transfer between those two cells. We can also take a kind of genetic approach to this, a genome approach, and so we can look at what are the factors, what are the genes, what are the factors that regulate the genes we want to be regulating. So we can kind of draw a genomic network, and the genomic network has genes which regulate other genes, and those regulations cascade depending on their connections in the network. Okay? And so let's say that this is MyoD at the center there. And so you want to know which gene it is. Which gene it is will give you the strongest control over all the genes that you want to hit. But what you don't want to do is regulate the wrong genes. So we can visualize the genome and therefore the transcription factors that we should be getting as a circle. And this is the genomic space. This is the genomic space that we want to regulate. And then within this genomic space that we want to regulate, we can visualize transcription factors that control specific genes. So we might have a transcription factor 1, which regulates this area, number 2, number 3, number 4, number 5. And in this example, transcription factor 5 regulates a relatively small area of the genomic space that we're trying to regulate. But that's okay. That's okay, because it's the only way to hit this specific area of the genome that we're trying to target. Now, of course, this space, this circular space that we're visualizing, is a representation of all of the genes that we need to transcribe, to reprogram one cell to another cell. And so you could actually have some of these transcription factors kind of going out of the space that we want. And we want to kind of obviously avoid that because you might get untoward outcomes, but it might be the only way to get this space at the inclusion of this space, as an example. So the app, if you want to play with this, if you want to give this a go, is called Mogrify. Mogrify is pretty cool. And Mogrify will take the two cell types that you give it, and it will give you a number of transcription factors that will allow you to take cell number 1 to cell number 2.